In my laboratory, we are focusing mostly on rodent studies. We are using both rats and mice, but we have also human research, let's say. So I will try to show you a bit of everything. You had a very nice introduction of empathy and emotional contagion during Christian's talk. So I will be brief during my introduction. I would only I want only to tell you here that we, as well as other primates, are very good at recognizing emotions of others, both the negative and positive ones, and we readily respond to emotions of others. Usually, such abilities are considered to be important for successful social interactions. So we have to discriminate, recognize emotions of others to respond in an appropriate way, to build relationships with others. But what I would like to stress here is that reading emotions of others provide us also with important information about our environment, about threats in the environment, but also about opportunities, about safety of this environment. So in our studies, we focus a lot on what is actually the meaning of information, how this information is processed by the brain, and how it can be used to adapt to the environment. And the studies of recent, let's say, 20 years have shown that also rodents have a very high abilities in recognizing and responding to emotions of others, both rats and mice. Does it work? No. I will try to use this cursor. Okay, so this part I can skip, I think. You already heard that what emotional contagion is. It's automatic mirroring of the emotional state of another individual. And it's the very basic form of empathy. As I said, rodents are very good at recognizing emotions of others. It has been shown in very nice, very simple paradigm developed in Francesco Papaleo's lab, in which a mouse has a choice between two other conspecifics. One of them is emotionally neutral, and another is emotionally aroused. And the emotionally aroused here means that it's either restrained, so it is stressed, or is offered water after a period of water deprivation. So it is a relief, so it's considered to be in a positive emotional state. And in both cases, the freely moving mice, which has a choice between these two conspecifics, actually prefer to stay close to the emotionally aroused one. So they are apparently interested. But why is it so? How does this socially transmitted information influence individual behavior, how the brain processes such information, and how can it be used to adapt to the environment, to localize the threats in the environment, or to localize, for example, food or other opportunities in the environment. So this is what we are focusing on. And in the first part of my talk, I would like to focus on negative emotions. I will try to be brief to save some time for positive emotions, on which we are focusing now in the lab. And I'd like to tell you a bit more about it. So the story started quite a long time ago when I was a PhD student. And I noticed that when a rat that was subjected to fear conditioning is going back to its home cage, its partner that is naive, that was waiting the whole time in this cage, is getting apparently aroused. So there's what we observed is that this naive rat spending its time in the home cage after social interaction with a fear conditioned rat is exploring the environment, it's rearing, it's sniffing its partner, it's apparently agitated. So then we decided to look at it closer. We confirmed this risk assessment behavior, but we also looked into the brain. And what we found in the brain is the activation of the amygdala, the structure that is crucial for fear processing. And we looked at different parts of the amygdala, especially at the basolateral and central nuclei of the amygdala that are traditionally linked to fear conditioning. And we found that they are activated to the same level as in the rats that were subjected to strong fear conditioning. They got 10 food shocks. So just observing, just interacting with a partner that was fear conditioned actually caused very strong activation of the amygdala. So this inspired us to look closer into it and to check whether this information that is conveyed during social interaction is accurate and how the rats can use it. So we used the model that I just described, in which one animal is subjected to fear conditioning in a separate room. So we called this model remote threat model, because this is similar to the situation in which some threat, like a predator, is far away. So there is a threat in the environment, but it's rather remote. And we compared it with the model that was already described by Christian, that we called imminent threat model, in which our observer rat is witnessing fear conditioning of the partner. So the partner is just behind the perforated partition. So the observer can smell, can hear, can see the demonstrator. So it's similar to the situation when the animal is attacked by the predator. The danger is really close. So in these two models, we see completely different pattern of behavior of the observers. In the imminent threat model, we observe mostly freezing, as you could see during Christian's talk. In the remote threat model, we observe risk assessment behavior, rearing, exploration. So there are different behavioral patterns. But what about the brain? So when we look closer into the amygdala, we found different neuronal circuits that are activated in these two models. So we focus on the central amygdala, which is the output from the structure coordinating behavioral responses. And we identify two circuits by their connectivity and by the molecular markers that they express that don't overlap. And on the next day, we were able to reactivate these circuits in a novel environment. So we put our rats to the novel environment that had a hiding box, and one of the corners of the box was brightly lit. So rats are a bit afraid of brightly lit areas. And we measured exploration when we stimulated these cells. And what we found is that when we stimulate these imminent threat cells, the rats spend the time mostly in the hiding box, so they avoid exploration. But when we stimulated the remote threat cells, they explored. It was cautious exploration. We observed this stretch attent posture, so they showed some risk assessment behavior. So it seems that the information that they got during social interaction actually activated the circuits that then reactivated, helped them to adapt to the environment. So their behavior were appropriate when we think about what happened to the demonstrators. So it seems that the information that they get during social interaction is actually very accurate. Okay, so now let's look what is going on in our brain when we observe others being food shocked. So Christian showed you experiments involving pain. We are focusing here mostly on fear. It seems that there is a difference between these two kinds of the types of stimulation. So together with Andreas Olsson from Karolinska Institute, we developed the paradigm, or we modified the paradigm that was developed earlier by Andreas to be able to study friends or strangers that observe fear conditioning of their partners. So we invited to the lab pairs of friends. One of them was lying in an fMRI scanner, and another was our demonstrator, so was subjected to fear conditioning. And fear conditioning was performed this way, that the demonstrators observe computer screens on which they had squares, either blue or yellow ones, and one of them was associated with a shock to a forearm. So it was very similar to what we do to our rats. And the just observation of this field conditioning activated a set of structures in the brain, including anterior cingulate cortex, anterior insula, and the amygdala. This is exactly what we expected. And based on the previous experiments with pain contagion, we expected that this response would be stronger in the observers that are related, emotionally related to the demonstrators. So to verify this, we tested strangers. So we used the same movies, but now the people who watched them didn't know the demonstrators. And to our surprise, we found that the level of activation of the brain and the pattern of activation is exactly the same. We also measured the physiological responses, and they were as strong as in the case of friends. So it suggests that it's not so much about social relationship, but it's more about warning, about danger. So it's like when we think about it, it doesn't matter who is warning us. If the danger is around, the info about the danger is valuable regardless of the source of information. So this is what we found here. And then we looked also closer into the amygdala to check whether the two parts that are related to field conditioning are activated here. Yes, they are. Both basolateral and central-medial amygdala are activated. So in the next step, we planned a bit crazy experiment in which we checked whether the interspecific transfer is possible. So we checked whether field-conditioned humans can transfer the arousal, negative arousal, to rats. So the experiment looked like this. We had the experimenters. They were well-trained experimenters, so they knew what they are doing. They handled rats for several days, and so the rats knew them. And after several days, rats are quite happy to interact with the experimenter. So in the control conditions, we observed a lot of interactions between rats and experimenters. But when we subjected the same experimenters to field conditioning and then tested interaction between a rat and a human experimenter, the rats actually try to avoid the human hand. They spend most of the time in the part of the cage quite far, as far away as possible from the human hand. And this effect was short-lasting, but highly significant. And then we look at the activation of the amygdala of the rats. And again, the basolateral and central amygdala, crucial for field conditioning, were activated here. So it seems that this transfer between species is also possible. And when we compare the brain structures involved in this field transfer in human rat condition and human condition, it's very similar. The same parts of the brain are activated. So to summarize this part, what we found is this transfer between rats, between humans, and between rat and human is possible. So this actually raises the question that was mentioned by Christian, that maybe it suggests that maybe emotional discrimination and contagion could evolve primarily as a mechanism to gather critical environmental information instead than being a byproduct of mammalian parental care. Something to think about. Okay, so now I'd like to switch to more positive emotions. So what we are focusing on right now are positive emotions in rats and mice. I agree that it's more challenging. It's more challenging to design the experiment. But what I can tell is that both rats and mice respond very readily to positive emotional states. So we are focusing on how socially transmitted reward information influence individual behavior of mice. And then I will show you briefly the project in which we are focusing on the ability of rodents to transfer information about food location just by social interaction with a fed conspecific. So let's start with the first project in which we tested the behavior of group housed mice, and we used a system that we have developed to track social interactions of group housed animals that we call EcoHab. It allows us to, first of all, to look at the whole group of animals in a semi-naturalistic settings, and also to track the social hierarchy and check how social hierarchy affects responses in which we are interested in. So the system consists of 4 cages connected with the corridors. Each corridor is equipped with RFID antennas, and each mouse has a transponder with a unique number, so they are individually recognized. We know when they go through the corridors, and because we have 2 antennas, we know the direction. There is food and water available, so this is a home cage. They can live here for a long time. In other 2 cages, there are perforated partitions behind which we can present some stimuli, social and non-social stimuli. We can compare how much they are interested in different types of stimuli. This was inspired by how mice live in the natural conditions. They love digging tunnels, and along tunnels they build nests. So we have nests or cages and tunnels. So here I will show you the movie. The mice are quite happy in the system. This is the newer version, which is also combined with deep lab cut, so we can track the different body parts of the animals and track the details of social interactions in the cages. So not only the presence in the corridors. And we usually measure in the system interest in social stimuli presented behind the perforated partitions, how much time they spend together in the cages of the system, and how often they chase each other in the corridors. I will go back to it in a moment. So I'd like to show you the result of the project carried out by my former PhD students, Maciej Winiarski and Alicja Puścian, who looked at how socially transmitted reward information spreads among mice in the IntelliCage system, in the EcoHab system, sorry. So what they did is that they put the group of 10 mice into the EcoHab, and after the period of adaptation, they removed 2 of them to 2 separate cages, and one of them was offered 10% sucrose solution, which is highly rewarding. They love it. And another mouse drank just tap water. Then what they did, they put the bedding collected after 24 hours from the single cages behind the perforated partitions in 2 cages of the EcoHab. And they compared how much time the animals spent close to the stimulus. And what they found is that the mice are very interested in spending time and exploration of the smell of a rewarded mouse, much more than the smell of the mouse that drank just tap water. And this interest was pretty long lasting. It was observed for about 12 hours. And it didn't affect the general exploratory activity, so it seems that it was focused on this cage that contained reward smell. Okay, so in the next step, we decided to look at the social hierarchy influence, or the role of social hierarchy in this response that we've seen. As I mentioned, in the EcoHab, because we have tunnels, they motivate mice to chase each other. And the chasing rate is correlated with other tests of social hierarchy. So it's a very nice proxy we measure this behavior in an automated way, and we can tell by the rate of chasing, that is like one mouse following closely another. It's very fast behaviors, usually under 1 second. So we can measure how many times they chase each other, and we can represent it as a size of the circle shown here. And the lines show who is chasing who. And when we look at this chasing rate over days, if nothing happens, if the environment of mice is stable, this structure is also stable. They need about 24 hours to stabilize the structure, but then it is stable. Here I'm showing the first 4 days, but we measured it for like up to 1 month, and this remains stable. And it's correlated with, for example, with tube test, which is another measure of social hierarchy. Okay, so we use this feature of the system to check what happens when we introduce the smell of a rewarded mouse. And what we found is that there is a clear difference in response of mice depending on social hierarchy. Those who are at the top of social hierarchy, the dominant mice, respond most. They increase their chasing behavior. And why they do this? Probably this is to show who they are, that when they find the reward, they should be the first there. But also to gain some information, because chasing others in the tubes of the system, they have access to olfactory cues from the anogenital area that are likely very important to convey information about reward. I will show you later why I think so. So we confirmed that statistically that those who contribute to this effect are mostly the dominant animals. And then we make the situation a bit complex for mice. We used 2 ecoHEP systems. The group of mice lived in one of them, then 2 scout mice were transferred to another system in which they drink tap water and sweet water in 2 different cages. Then we removed the scout mice, we removed the bottles, we replaced them with tap water, so the olfactory traces were left in the environment, but now the mice had access only to tap water. And what we found is that and other mice moved into the environment. So we tested their response. We've confirmed what we showed before. They preferred the part of the cage where before the scout mice found sweet water. They also preferred drinking from this bottle, even if now it contained only tap water. And again, we observed this influence of social hierarchy, with the dominant animals contributing most to the change. So in the next step, we check the role of the prolimbic cortex. And we focus on the prolimbic cortex because it was shown before to be important to recognize social stimuli, for example, by Francesco Papaleo's group, the very simple protocol that I showed you at the beginning. So we wanted to see what happens when we manipulate the plasticity and activity of the prolimbic cortex. And to manipulate the plasticity, we used nanoparticles that gradually, when injected to the brain, released TIMP1. TIMP1 is an inhibitor of matrix metalloproteinase 9, which is an enzyme important for synaptic plasticity. The previous studies showed that the TIMP1, when injected to the prefrontal cortex, blocks LTP. So there is a proof that it affects plasticity there. And we injected our mice with the nanoparticles. So for about 5 days, we expected to have reduced plasticity in the prolimbic cortex. And we tested the same mice before and after injection. And what we found is that injecting them, so blocking the plasticity in the prelimbic cortex, leads to decreased interest in social stimuli, signaling a reward, but also destroy the social structure. The mice actually almost stop chasing each other, and they decrease the activity. We here affected both measures, both the response to reward at the level of social structure and the direct interest in the social reward. So then we decided to try to distinguish between these two responses. And what we did, we blocked the activity of pyramidal neurons in the prolimbic cortex using chemogenetic approach, SAM method, that gives you inhibition for about 2 hours. And when we did it, what we observed is the decrease in preference, but the social structure was intact. So it seems that the prolimbic cortex is important for both the direct response or interest in social reward and in the maintaining of social structure. But for maintaining of social structure, the plasticity in the prolimbic cortex is necessary. So to briefly summarize what I showed you in this part is that mice explore scent cues that are left by other animals in the environment, the other animals that found a reward, and they can tell the difference between neutral and reward-related smells. That these scent cues help mice to find rewards in new places. And the response to the presence of a reward smell in the environment is influenced by the position in social structure. Those responses are modulated by the prolimbic cortex. Okay, so in the last part, I would like to focus on the experiments that aim at testing whether mice and rats are able to transfer information about food location. So here we decided to go back to a simpler behavioral protocol. It was the project carried out by my former PhD student Mateusz Kostecki, who designed this behavioral protocol. The protocol looked like this. The animals were kept in pairs, so they knew each other, they were familiar. They were exposed for 3 days to the 3-chambered apparatus. And 2 side cages of this apparatus were distinguished by different visual stimuli and different smells. And then the mice, during the test days, during the test day, the donor of information was fed in the experimental cage. So in one of the compartments, the side compartments, it was offered regular food. When the food was eaten, the donor was transferred to another compartment. So the idea was that it has a smell and exposure to both compartments. And then the mice interacted in the middle compartment of the cage. The side compartments were cleaned, and then we tested the preference of the naive observer for the compartments. What we did here is that we offered food to the donor in the cage, the side of the cage that was less preferred by the observer during the habituation phase, so we expected change of preference during the test phase. In the control group, the donors were offered food just before being introduced to the experimental cage. So they ate food in the home cage, so they were exposed to all stimuli, but the stimuli were not connected to each other. So the experimental group is called food plus location, and the control group food only. And what we found is that when we look at the preference, the control group, nothing changed, but in the experimental group, there was a clear change in preference. The observer mice started to prefer the compartment in which the demonstrators were fed. So we clean the cage, but we can't be sure that some traces of our food are not left there. So we decided to control for this, omitting the social interaction. And when we did it, the effect disappeared. So it seems that social interaction is important for this. So then we check what happens in rats. Again, control group, nothing. Experimental group, there is a clear change in the preference. So they start to prefer the cage in which the donor of information was fed. For rats, it was even more difficult because they interacted not in the experimental cage, but outside. So it was a bit more challenging, but still they were able to do it. We look what is, because this looks a bit like magic, so we looked at what they are doing during interaction, trying to figure out how they can do it. So we look at the sniffing behavior. They involved a lot in sniffing. The nose-to-nose sniffing, that is very intense, but it's not different between the groups. But also in anogenital sniffing, that it's more intense in the experimental group. So it seems that some olfactory cues released in this area may be important here. Then we measured ultrasonic vocalizations. They almost don't vocalize, so it seems that it's not it's not the likely channel of communication here. Then we thought that, okay, maybe they show where the food was just by spending more time close to the compartment where the food was offered to the donor. But it seems not to be the case. We firstly measured it manually, then we trained the algorithm to, like, using machine learning approach to be more objective. It doesn't work. So it seems that it's more supple. So then we look into the brain, and we decided to look at the spatial maps. The idea was that they have to somehow represent the space, and maybe this representation is involved during social interaction. So to look at this, we use one-photon miniscopes to measure activity of CA1 hippocampal cells. And we measured the activity during the last habituation session, during social interaction, and then during test during which the mice explored the cage, and we measured preference for side cages. And what we found that already during interaction, when we compared the groups, in our experimental groups, more cells representing the target cage, that is the cage in which the donor was fed, are activated. When we quantify the activity of the cells, we found the differences between the groups. So in the experimental group, which is the bottom panel, there was the decrease of activity of the cells representing non-target cells, non-target compartment, and increase of activity of the cells representing target compartment. So again, more activity related to the place where the donors found food. We look also at the stability of cells over days. It's a memory measure. So if they memorize if our observers would memorize what happened to the demonstrators, we should expect increased stability. It was not the case, but maybe it's not surprising because they were actually not finding food. I mean, the observers were informed that the food is there, but they were not fed themselves. So it's no point in learning about this place. So it shows that it's a kind of mental representation during social interaction, the mental representation of the space in which the donor was fed. And the last thing that I would like to show you is the global brain activity, because at this point we didn't know exactly what to look at. We decided to look at the activity of the whole brain. Using IDISCO method and CFOS activation as a measure of activation of neurons. And we measured the activation of the brain after social interaction. So what we found, there is like a long list of structures. It's not very readable, but I try to summarize the main results here. What we found is that there is a high asymmetry of the activation in both groups. So in the experimental group, what we see is the strong activation of the higher sensory cortices and orbitofrontal cortex. But in the control group, there are many structures related to memory, arousal, emotions, motivation, activated also the primary sensory cortex. So it seems that it's like a sort of a change in navigation strategy. In the experimental group is more related to the sensory cues they get from the donors that were fed. And then we decided to check what happens when we actually inhibit the memory that was formed on the previous days, because we observed in the control group that they activate the memory system a lot. So we decided to inhibit the pathway from the ventral hippocampus to anterior olfactory nucleus that is important for episodic memory, olfactory-based episodic memory. And what we found, that when we do it, we actually help our observers from the experimental group. When this pathway is inhibited during the first 5 minutes of exploration, you can see the increase of preference. So it seems that like inhibiting of the memory probably gives them better access, or there is a higher influence of the sensory information that they get from the donors of information. So to summarize this part, we found that both rats and mice can learn about food locations from informed peers. Single photon imaging shows that hippocampal contextual cells are reactivated during social interaction. This suggests mental simulation of others' experiences during social interaction. And the whole brain CFOS mapping shows a shift from memory-based to sensory cue-based navigation during social interaction with an informed peer. So I hope you convince you that the rodents are interested in positively aroused others, and that they can get some information from such interactions. Here I would like to thank many people from my laboratory who were involved in the studies that are the results of which I showed you today, and also our collaborators. I tried to name the most important people during my presentation. Thank you very much for your attention.